Assalamu alaikum friends. This is Ajaz Ahmad and welcome to my Doctv. A typical cell consists of a plasma membrane separating the inner contents or cytoplasm from the environment around the cell. Cytosol is the fluid component of the cytoplasm. The cytoplasm includes the cytosol and all the organelles and structures in it. The nucleus is not considered to be a part of cytoplasm. Organelles perform essential cellular functions. The nucleus is the largest organelle and it contains the cell's genetic information encoded in the molecules of DNA. Plasma membrane, cytoplasm, organelles, cytosol, nucleus. Now comes to the structure of the plasma membrane. The plasma membrane encloses the cell's cytoplasm. It is made up of two layers of lipid molecules known as the lipid bilayer. The hydrophilic head of the lipids, which are attracted to water, face outward towards the fluid inside and outside the cell. The tail of lipids, which are hydrophobic, face inward towards each other. Proteins in and around the lipid bilayer form ion channels, carry substance through the membrane, act as a marker or receptors, and facilitate other cellular functions. Passive and active transport. Substances such as nutrients, gases, and waste are constantly moving into and out of cells. When the concentration of substance is higher on one side of the cell's selectively permeable membrane, certain molecules may move by osmosis or diffusion through the membrane without the cell using any energy. This process is called passive transport. When the cell needs to move molecule from an area of low concentration to one of higher concentration, it uses specialized channels or carries in the cell membrane. This process expands energy and is called active transport. Transport vesicles. Some substances are transported to and from the plasma membrane in membranous sacs called transport vesicles. This process expands energy, so it is a form of active transport. Plasma membrane, transport vesicles. Osmosis. Diffusion of water across the cell's plasma membrane is called osmosis. The direction of osmosis depends on the concentration of the solution around the cell. In a hypertonic solution, there is a greater concentration of water inside the cell than outside it. Water flows out and the cell shrinks. In an isotonic solution, like normal blood, the intake and output of water is balanced and the cell functions correctly. In a hypertonic solution, there is a greater concentration of water outside the cell than inside it. The cell swells and may even burst as water flow in. Organelles. Organelles within the cell perform essential cellular functions. Organelles, cytoplasm, cytoskeleton, and centrosome. The cytoskeleton consists of proteins called microfilaments and microtubules that supports the cell. The centrosome contains centrioles and pericentriolar material that are essential for cell division. Cytoskeleton, microfilaments, microtubules, centrosome, centrioles, pericentriolar material. Endoplasmic reticulum. The endoplasmic reticulum is a folded membrane network that synthesizes proteins and other substances. Rough endoplasmic reticulum contains ribosomes, but smooth endoplasmic reticulum does not. So we have endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum. Golgi complex. Proteins produced in the endoplasmic reticulum are processed in a set of membrane sacs known as Golgi complex before being released. Endoplasmic reticulum, Golgi complex. Lysosomes and peroxisomes. Lysosomes are capsules inside the cell that contains digestive enzymes. Their function is to break down substance consumed by the cell or the recycle waste. Similar organelles called peroxisomes process and neutralize toxins, metabolize long chain fatty acids, and contribute to energy production. Peroxisomes. Nucleus. The cell's nucleus contains its genetic information encoded in the molecules of DNA, which are arranged in bundles called chromosomes. The nucleus is separated from the cytosol by a membrane. The nuclear envelope inside the nucleus, a central body called nucleolus, produces ribosomes. Nucleus, DNA in chromosomes, nuclear envelope, nucleolus. Mitochondria. Mitochondria use oxygen taken in by the cell to produce ATP, the molecule that cells use as fuel for energy. Mitochondria. Ribosomes. Ribosomes synthesize proteins. Some ribosomes float freely in the cytosol, while others are attached to cellular structures. Free ribosomes, attached ribosomes. Cellular respiration definition. Cellular respiration is a series of processes that take place within a cell to break down glucose to make adenosine triphosphate. The three main processes are glycolysis, the citric acid cycle, and oxidative phosphorylation electron transport chain. Glycolysis starts in the cytosol. The rest of the process occurs in the mitochondria. Cellular respiration. Cellular respiration comprises three main steps. It begins when glycolysis breaks down a 6-carbon glucose molecule, producing 2,3-carbon pyruvic acid molecules and a net gain of 2 ATP molecules, which the body uses as cellular energy. High energy electrons are released. Each 3-carbon pyruvic acid molecules enters the mitochondria and then enters the citric acid cycle. The second step, 2 more ATPs are created and carbon dioxide is released as a waste product. High energy electrons are again released. Third and final step, oxidative phosphorylation, involves the electron transport chain and chemiosmosis in the inner mitochondrial membrane. It is during this phase most of the ATP molecules are created. The three steps of cellular respiration can produce up to 38 ATP molecules from one glucose molecule.
Proteins. Proteins are chains of amino acids that have many functions in the body, including acting as hormones in the endocrine system, contracting muscles, and transporting substances in the bloodstream and across the cell membranes.
Protein synthesis. The information in DNA in the nucleus is used to produce proteins. The assembly of proteins amino acid chain occurs in the cytoplasm and is facilitated by the ribosomes. DNA in chromosomes, nucleus, cytoplasm, ribosomes. Transcriptions. Definition. Proteins are made from the template of a cell's genetic code through the process of transcription and translation. In transcription, the double helix DNA molecule is unzipped and a messenger RNA, that is mRNA molecule, is created from the DNA template. All cellular protein building and much of the cellular gene expression occurs through the two principles called transcription and translation. In transcription, DNA information is encoded into RNA for genes that code for protein. The RNA is called messenger RNA. The process begins when RNA polymer opens the double helix of DNA like the slider on a zipper. It unwinds the DNA double helix and places the two DNA strands apart. As the RNA polymer continues down the length of the DNA, several actions take place. It reads one strand of the DNA and transcribes the DNA's information. It creates an mRNA version of the DNA. The mRNA transcript is a complementary copy of the DNA sequence. When the transcription ends and the mRNA transcript is complete, it separates from the DNA templates and it exits the nucleus through a pore. The transcript enters on its own with the next step, the process of polypeptide synthesis translation. Translation definition. Translation is the formation of a linear chain of amino acid using the transcript information provided by an mRNA strand. During translation, the mRNA template is decoded or translated. to synthesize a protein within the ribosome, a 3-base sequence in the mRNA that codes for a specific amino acid in the protein is called codon. Each codon specifically codes for one of the 20 amino acids found in the body. Codons and the ribosome. The ribosome sits over 2 codons of mRNA at a time. Each codon sits in a specific pocket of the ribosome. During translation, these pockets allow transfers RNA, that is tRNA molecule, to match their anticodon up to the mRNA codon. When there is a tRNA in each pocket, the ribosome catalyzes formation of a peptide bond between 2 amino acids on the tRNAs. Translation. Translation begins when the mRNA enters the cytoplasm and a small ribosomal subunit attaches itself to the end of the mRNA strand. At the same time, transfer RNA molecule recognizes and binds to the start codon on the mRNA strand. tRNA recognizes the mRNA codon through its anticodon, which has sequence complementary to the mRNA codon. It is the tRNA molecule that carries the amino acids building blocks. It is the tRNA molecule that carries the amino acid building blocks of the protein. Next, a large ribosomal subunit joins and forms the functional ribosome that will join the amino acids on the tRNAs to synthesize the protein. When the amino acids of the 2 tRNA molecules are joined, the tRNA in the first position is released to get a new amino acid. Growing protein is attached to the tRNA in the second position. That mRNA is then pulled through the ribosomes so that the tRNA within the protein chain is now in the first position. Now a new tRNA with the proper anticodon can come into the ribosome at the open position. This pattern continues as tRNA after tRNA bind to the mRNA codons and their amino acids are added to the growing protein chain. Towards the end of the mRNA sequence, the ribosomes get to a stop codon, which does not code for any amino acid. This signal end of the protein and the translation process is complete. If you are enjoying my classes, please subscribe it. If you want anything detailed about anatomy and physiology, please comment me. Thank you. Stay safe. Have a healthy life. Khuda Hafiz.